What is your name and where are you from?
My name is Sarah Nehaus and I'm from North Florida.
What are you doing here in Panama on Barrow, Colorado Island?
I'm a PhD student at the University of Cambridge in England, and this is my field site.
So what is your big question?
My big question is, well, I need to give a little bit of background to explain my question. Under elevated carbon dioxide conditions, trees are expected to produce more leaf litter, and this has been shown to occur in temperate forests. And if the same occurs in tropical forests, there's going to be extra leaf litter on the ground. And my question is, how does this extra leaf litter, if it does occur, how will this influence carbon and nutrient cycling in tropical rainforests?
So here in Gigante, what is the question that you're studying?
So our question is, how important is leaf litter for carbon and nutrient cycling in tropical rainforests? And the reason we're asking this question is because there is 20 to 25% of terrestrial carbon stores and nearly 30% of soil carbon stores are found in the tropics. So there's a lot of carbon stored in the tropics, and changes to carbon cycling in the tropics could be important globally if there are changes occurring. Other experiments have showed that elevated levels of carbon dioxide cause trees to produce and drop more leaves. So there's a bigger input of leaf litter under elevated carbon dioxide conditions. So what we have going on here is research plots at the ecosystem scale, and these over here are the litter removal plots where the leaf litter is raked up once a month and it's transferred to the litter addition plots, which this plot over here is a litter addition plot.
And so what is your hypothesis?
Well, because we're transferring, there's two different questions at play here. There's the nutrient cycling and the carbon cycling. For nutrients, we would guess that tropical soils are typically poor. They're older soils. A lot of soils in temperate forests have rocks, and as rocks weather, they release nutrients, but these soils are old and are fairly weathered as they are. So the main source of nutrients here is leaf litter and decomposing organic material. So our guess is that when we remove all of this leaf litter, that we would expect to see the forest suffering from nutrient deficiency. And we would expect in the addition plots that maybe we have a surplus of nutrients.
What observations are you making?
I'm trying to develop carbon budgets for the different leaf litter manipulation plots. My observations that I'm making are that I'm measuring carbon amounts at different places throughout this cycle, and I'm doing this over time.
And how do you collect your data?
Well, all of the different pieces I collect in different ways. I'll give you an example of two of the collections that have already started. One is I collect litter fall, which is the dead leaves that have fallen from the trees to the ground, but I intercept them before they make it to the ground with a square trap made with PVC legs and screen net in the trap to catch leaves before they make it to the ground. And I collect all the leaf litter in these traps once a month, and then I dry the leaf litter and weigh it. And then twice a year I collect this leaf litter and grind it up and send it to a laboratory for nutrient analysis. So the second kind of sample that I'll explain is that I collect leaf litter standing crop samples. And leaf litter standing crop is the leaf litter that has its leaves that have fallen from the trees to the ground but have not yet decomposed. And how I do this is by putting a metal disc that is a circle, it's actually a pizza crisper, on the floor of the forest in a random location, and then I cut around this disc and then collect all of the leaf litter that is in the circle underneath the disc. I then take this leaf litter back to the lab where I wash it and I sort it to determine which part of my sample falls within the definition of leaf litter standing crop, because there's a continuum on the forest floor from fresh leaves to fully decomposed leaves. Once I've washed the sample and sorted it, then I dry it and weigh it. And I will probably send samples twice a year away for nutrient analysis. The reason that I do nutrient analysis 2 times a year is because this forest has 2 seasons, the wet season and the dry season. So I would send one sample away for nutrient analysis mid-wet season and another one mid-dry season. Okay, so first before we get close to the ground, let's make sure we're not getting too close to a snake. And this looks like this place is safe. Okay, so this is the spot that we measured off, and here it is.
That's it just came out. I don't know what you want to do about that.
Oh, it was under.
Yeah.
Well, that's alive. See how it's green?
Okay, so you don't want
So we don't want that.
Okay.
So. Wow. It's a big sample. Okay, so now we can take this part out because it has too big of a diameter. It's not included. I'm going to take all the leaves off of it.
We don't want you. Oops. Yeah, what?
So if you're
down, yeah.
if you collect extra, that's no problem because we'll wash away the extra in the lab. So and in a sample like this, there's sort of this mixture of leaf fragments and humus, and the layers are not as defined. They're really mixed together. So it's better to collect more, and then we'll just wash the extra away in the lab. So you see when you get to this point, you have no idea where the circle is. And we had to right by that. So that's good.
Okay.
And now we put a flag in the middle to be sure that we don't sample in the same place again. And here's the case for the knife. And I'll write down which bag number we used. This is bag number 341. Okay, so now we're in the lab, and here's our samples from the refrigerator. So the first thing we're going to do, pick one of the samples, and our sample LSC1, sample 11-3. And it's easier, I found, to do a little bit at a time, because if we dump all this in there, it's just a mess. So we'll start a handful here. It's a good amount to start. I'm going to come around and look at this. So this is what we collected, and you can see, like, for example, this leaf has soil on it. And there's soil that's falling to the bottom here. And that's what so there's two things we're trying to do here. One is wash the soil off of, like this had a lot of soil on it. Wash the soil off of the sample because soil is a lot heavier than dried leaves and twigs. So that would greatly influence the mass of our sample, and we're looking for the mass of the litter, so the leaf litter. Okay, so for, and roots, we're not collecting roots. And if you see the pieces attached to the roots, these are all less than 1 centimeter, so we're not collecting any of that. For twigs, they need to be at least 1 centimeter long. This one's clearly long enough. So this we would collect, but I'm gonna just rub the soil off a little bit. And let's see if maybe this is too big. And so the other criteria for twigs is that its diameter cannot be more than 1 centimeter. So we're going to check here, and this is right at 1 centimeter. So this, because this width is the 1 centimeter. So this we would collect, but if it's wider than this, we're not going to collect it. So this. Okay, so now that we've taken out the big pieces, and if you want to come take a look at this, it's some muddy water with little stuff floating in it. So now is when we use this tool, and you get just, you don't want to overfill it or the smaller things can't escape. You sort of shake it around like this. So this is accurate enough for what the detection limits are, and if you look with this, you can see that it actually does a fairly accurate job. It's not 100% perfect, but it is really close to only collecting things that are the right size.
Do you always find what you expected to happen? What happens if you don't find the answer to the question you ask?
Actually, in this long-term experiment, we haven't found what we expected, and that's the most interesting thing that can happen sometimes, because if all of the assumptions right now point towards one result and we get something totally different, that's very interesting. That means that maybe we should look back at some of our assumptions that we've made and some things that we thought were true, and that in a lot of ways is the most interesting outcome, is to get something completely different.
So why is your research important? Does it contribute to a better understanding of global change?
Well, yes. My research is trying to understand how tropical forests will react to elevated carbon dioxide conditions. So my research does directly relate to global change. In my research, we have 45 meter by 45 meter plots where we have simulated an amplified version of the leaf litter conditions that are expected to occur under elevated carbon dioxide conditions. So within this context, we're able to study a simulated forest that has already experienced climate change. It's an existing forest, but we've simulated some of the effects of climate change in this forest.
What makes a good question for research?
I think that there's a lot of attributes to a good question. Of course, it's best if it's something that's not already answered. I think that a good question is a question that the answer is interesting to find out. And also, I think that it's important that the researcher is interested in the question, because then the work will be better, because a lot of times science can be very tedious. And if you're excited about the outcome, it makes it easier to make it through the tedious days.